and thanks to the organizing committee for inviting me. Here's a plan for my today's presentation. First, we'll look at how magnetic forces affect cell membrane potential. Then we'll dive into how magnetic fields influence skeletal muscle cells. We'll explore the effects of a static magnetic field on DNA synthesis, and then I'll demonstrate how magnetic fields destroy the molecular mortar inside breast cancer cells in mice. We'll round off with ultra-high static magnetic fields' impact on diabetic and depressed mice. Let me start with a quick quiz. What happens if you put a magnet near a mouse? Right, the mouse is diamagnetic and therefore it gets pushed away with force F. The next question, what will happen if we make the magnet bigger while preserving its magnetic characteristics? Let me answer. The magnetic force will disappear because it comes from the magnetic gradient. When the magnet is much larger than the mouse, the magnetic field becomes practically uniform and the magnetic force zero. The next slide is about the forces at play within living cells. Let's compare their magnitudes to the intracellular forces driving the cell machinery. The first is the Lorentz force. The second is the magnetic gradient force, which is proportional to the gradient of the square of the magnetic induction B. The next is the so-called concentration gradient magnetic force, which is proportional to B squared and the gradient of the concentration of diamagnetic or paramagnetic species. Estimations show that the magnitudes of these magnetic forces lie in the range between 1 and 100 piconewtons. Note that the Coulomb forces driving ion channel gating are about 10 piconewtons. If we look inside a living cell, we will see that the cell is an electrical unit. So, in cells, many processes are controlled by the membrane potential, voltage, which typically ranges from -3 microvolts to -100 microvolts. Why is it important? Slide 6. Why is it important? Because the membrane potential distribution determines an embryo's growth. Thus, controlling the membrane potential enables us to achieve various phenotypes. For example, changes in the membrane potential by an electric field enables the growth of an organism with 4 eyes, as reported in the paper by Levine. Let's fantasize a little. By manipulating the membrane potential with magnetic fields, we could create a frog with 4 tails and 4 heads. It would be a monster, probably. But now let's turn our attention to a more serious question. Here you see the distribution of different types of cells across their membrane potentials. As you can see, cancer cells have very small membrane potentials. Namely, this enables them to be very plastic and invasive. In a similar fashion, stem cells also have small membrane potentials. In contrast, neurons, glial cells, and skeletal muscle cells have the highest membrane potentials, reaching up to -100 millivolts. This raises an interesting question. Can we use magnetic fields to alter the cell membrane potential? Could a magic magnetic scroll do this? We believe that the answer is yes. We demonstrate such a possibility for skeletal muscle cells. Slide 8. We applied a 70 millitesla AC magnetic field with a 1 to 10 Hz frequency to C2, C12 skeletal muscle cells. The setup scheme and special distribution of the magnetic fields are shown here. We observed an enhancement of cytosolic calcium in response to the application of the magnetic field. The underlying mechanism is as follows. The AC magnetic field induces an electrical field in cells. This electric field alters the membrane potentials by a few millivolts. The change in the membrane potential opens voltage-gated ion channels, leading to an increase in cytosolic calcium levels. I will show you the details on the next slide. Let's look at the calcium level as a function of time in the pulse regime of the magnetic field. When the field is switched on, the calcium level increases. When it is switched off, it returns to its initial level. The increase in calcium is significant and reaches 35% depending on the field's frequency and amplitude. These results hold significant potential for various medical applications, including improving skeletal muscle functions, in myopathies, pain relief, and anti-inflammatory effects. There are experimental results on DNA synthesis in 4 types of cancer cells in the presence of a 1 Tesla static magnetic field. The figure that we see on the screen displays the relative DNA synthesis rates in the upward and downward magnetic fields. They are counterintuitive. Indeed, DNA synthesis depends on the magnetic field direction. Specifically, for 2 types of cells, the upward magnetic field rather than the downward field significantly decreases DNA synthesis. To unravel this asymmetry, we must go into the origin of life and the homochirality of living molecules. Let's have a look at the chirality selection for life. At the molecular level, it begins with homochiral molecules of left-handed alanine, followed by the right-handed DNA helix. At the cellular level, there is a left-right asymmetry in cell division. Moving to the organism level, we observe the typical right form of the snail Fruticula lanci, which is more viable than the left form. Additionally, there's a left-right asymmetry in the human body. Intriguing, the human brain undergoes left-right inversion under the influence of a magnetic field. Now, let's go back to our experiments with DNA. As illustrated here, DNA carries negative charges due to phosphate groups in nucleotides. DNA rotates during its replication. Thus, the moving charges are subjected to the Lorentz force, which changes direction in response to the upward or downward magnetic field depicted here. As a result, a segment of DNA rotates with a higher angular velocity in the upward magnetic field than in the downward field, as described by this formula. This increase in DNA rotation speed leads to the generation of an additional number of supercoils. There is an analogy with the creation of supercoils in a phone cable. It's evident that DNA with a high density of supercoils replicates at a slower pace than DNA with a lower density of supercoils. This fact explains our in vitro experimental results with cancer cells. We performed in vitro and in vivo experiments in a 9.4 Tesla static magnetic field, and these are the most interesting results we obtained. Upward 9.4 Tesla static magnetic field treatment for 24 hours caused A549 DNA synthesis inhibition. It also significantly increased ROS and P53 levels and caused G2 cell cycle arrest. Exposure to the same magnitude upward static magnetic field for 88 hours significantly inhibited A549 tumor growth in mice. The diagram shows the mechanisms of actions of downward and upward magnetic fields. In the case of an upward 9.4 Tesla magnetic field, the supercoil density is high. DNA synthesis is sufficiently inhibited. This triggers the ROS level elevation and activation of the P53 protein, a key tumor suppressor. In mice, this causes a significant tumor inhibition. In contrast, in the downward magnetic field, DNA has a low density of supercoils. DNA synthesis is not inhibited. There are no ROS, no p53 protein, and therefore no significant effects on the tumor. Slides 14, 15, and 16. The proposed mechanism is valid when DNA is oriented, at least partially, parallel to the field direction. The magnetic properties of DNA, however, remain an open question that requires further investigation. Recent in vitro experiments, however, have shed some light on this matter already. In the next two slides, we discuss in vitro and in vivo experiments with cancer cells. We will demonstrate how a rotating magnetic field destroys the actin-myosin motor of breast cancer cells in 5 minutes. We used the magnetic system consisting of two rotating magnets with a frequency of 4.2 Hz. What is the mechanism of the breakdown of this molecular motor? The key factors are here. The top panel represents the sham experiment. This is for 0.1 Tesla magnetic field, and this one here is for 0.4 Tesla field after 5 and 20 minutes of treatment correspondingly. You can see that the cell shape and size, as well as F-actin filaments, were drastically changed by the magnetic field. On the right, the F-actin filament length decreased in both fields, 0.1 and 0.4 Tesla. The number of tumor metastasis nodules on the lung tissue also decreased. Slide 17. Now let's have a look at the mechanism. In this plot, you can see the integral signal from F-actin as a function of time in sham control cells and in the cells exposed to the rotating magnetic field, which means a periodical assembling and disassembling of F-actin. F-actin filaments assemble themselves from G-actins. Each G-actin has an electric dipole moment. A rotating magnetic field induces an electric field that orders the dipole moments of G-actins. So, without a magnetic field, G-actins assemble in F-actin filament-like manner that is shown in the figure on the screen. In case of the electric field, the dipoles with the same orientation can be incorporated into F-actin, but in this case, the GG dipole-dipole repulsion decreases the binding energy from 27.8 kilojoules per mole to 10.2 kilojoules per mole. So, in an F-actin filament, one can find a big domain, which we see in the figure as well. Finally, a rotating magnetic field can destroy the actin-myosin motor of breast cancer cells by breaking this weak domain. This mechanism also works in vivo. I will demonstrate it on the next slide. Slides 18 and 19. Here we demonstrate that a rotating magnetic field significantly reduces mouse breast cancer metastasis and extends mouse survival by up to 46%. Thus, the perturbations of F-actin inhibit breast cancer metastasis, having minimal effects on normal cells. These results open perspectives for magnetic treatments for metastatic cancer. And now, let's talk about the biological effects of ultra-high magnetic fields. Currently, in clinics, MRI scanners operate with magnetic fields between 2 and 9 Tesla. However, one should increase the field and its gradient values to reach the highest image resolution. Up-to-date MRI scanners that operate with magnetic fields of 21 Tesla are used in preclinical studies. So, of course, the question of safety of such huge magnetic fields immediately arises. This here is the world's largest superconducting magnet, which generates a static magnetic field up to 45.22 Tesla. And this is me standing next to it. Now let's discuss the experiments with diabetic mice exposed to static magnetic fields of magnitudes up to 9.4 Tesla for a period of 14 hours. There were 4 groups of mice in our experiments. The first group was made up of healthy mice. This was our control group. The second group consisted of mice with type 1 diabetes, the third one of mice with type 2 diabetes, and the fourth one of mice with severe diabetes. The experiments were performed in quasi-uniform and gradient magnetic fields with a gradient larger than 10 Tesla per minute. On the left, you can see the survival rate curves of healthy and all diabetic mice with or without magnetic field treatment. The black line represents healthy sham groups, none of which died during our experiments. The dark red line represents diabetic mice of all types after the high static magnetic field exposure. On the right, you can see that exposure to a quasi-uniform 9.4 Tesla magnetic field for 14 hours was safe for all 3 types of diabetic mice, but high magnetic fields between 1 and 8.6 Tesla with a gradient larger than 10 Tesla per minute were harmful, especially for mice with severe diabetes. The tentative mechanism underlying these effects is that exposure to high gradient magnetic fields increased the oxidative stress state in diabetic mice. In vitro cellular studies showed that high gradient magnetic fields increased cellular reactive oxygen species and induced apoptosis of MS1 cells. Last but not least, let's consider the antidepressive effect of static magnetic fields on mice. 84 healthy mice were divided into 3 groups: control, sham, and field. The latter were exposed to a 33 Tesla magnetic field for 1 hour. The timeline of the experiment is shown here. 1 week of adaptation, 1 hour of field exposure, and later on, various tests. After the field exposure, mice successfully passed all these tests. They were very active, very social, and happy. The positive outcomes of the tests and the antidepressant effects of the ultra-high magnetic fields can be attributed to the increased expression of both oxytocin and CFOS in the mice's brains. The analysis of brain tissue revealed that the ultra-high static magnetic fields increased the oxytocin level by 165% and the CFOS level by 36%. The CFOS protein is a marker for neuronal activity. We proposed the following tentative mechanism for the elevation of oxytocin. It is known that in the brain, oxytocin is generated by neurons under osmotic stimulation. The presence of an ultra-high magnetic field can exert additional pressure on the cell membrane due to the concentration gradient magnetic forces. Thus, in a high enough static magnetic field, the magnetic pressure may cause an imbalance in the osmotic and hydrostatic pressures, which in turn changes the fluxes of ions transported through the cell membrane and the osmotic pressure as well. We hypothesized that the change induced by the magnetic field and the hydrostatic and osmotic pressure balance can affect oxytocin levels through mechanosensitive ion channels. In summary, ultra-high static magnetic fields positively enhance emotions and social novelty. This is likely associated with increased levels of CFOS and oxytocin in the mice's brains. These findings suggest that ultra-high static magnetic fields hold potential as future treatment modalities for depression.